Welcome back to this next video, and this is actually the third part and the final part on the intrinsic apoptosis. In the last video I've told you that when the concentration of the BH3 only protein that increases because of the intrinsic signal like the DNA damage, the P53 is actually going for the transcription of the BH3 only protein family of the proteins, and their concentration is increasing. And when their concentration is increasing, they are actually going for the formation of the BH123 aggregates, and the BH123 aggregates are actually responsible for the release of the cytochrome C from the intermembrane space of the mitochondria. So when the cytochrome C that is released, that is going for the activation of the APF1, and when the APF1 that is activated after the binding of the cytochrome C, its card domain that has been exposed now, and the card domain, the 7-APF1 molecule, the 7-APF1 protein, they combine with each other, utilizing their card domain to make this big structure which is known as the apoptosome. And I've told you this apoptosome was a large quaternary protein structure which was responsible for the activation of the initiator caspases. I've told you that the initiator caspase that is in the intrinsic pathway is called as the Procaspase 9, and it have got three domains: a long pro domain, a large subunit, and the small subunit. And in the long pro domain you have got this card domain, and this card domain of the Procaspase 9 is actually interacting with the apoptosome, and when the Procaspase 9 utilize its card domain to interact with the apoptosome actually cut at these two sites. And when these two sites they are cut, the large and the small subunit they get separated from each other, and so the apoptosome is going for the cleavage of multiple copies of the Procaspase 9, utilizing giving you a large number of the large subunit and the small subunits. And two large subunits and two small subunits they actually combine with each other to give you an active caspase 9. When the initiator caspase 9 that has been activated, that is actually going to activate the effector or the executioner caspases, for example the caspase 3 or the caspase 7. This executioner or the effector caspases again have got three domains: the pro domain, the large subunit, and the small subunits. And the active caspase 9 is actually making cut at two sites, one at the intersection of the pro domain and the large subunit, and one at the intersection of the large and the small subunit, thereby separating the large and the small subunit from each other. And the active caspase 9 are going to cleave multiple copies of the executioner caspases, and just like the initiator caspases, you need two copies of the large subunit and two copies of the small subunits to make an active executioner caspase, for example an active caspase 3 or an active caspase 7. Now in this video I want to talk about the caspases. What are these caspases? So these caspases, they are actually a family of the protease enzymes, and they are very essential for the process of the apoptosis. Now these caspases, if you look at the name from where this term caspase come, so if you look at this C is actually for the cysteine, this ASP is actually for the aspartate, and this ASE show that this protein is enzyme in nature. So this is actually the cysteine aspartate proteases, or they are the cysteine aspartases. But these two names, they are not giving you any clear idea why they are called as the caspases. This third name, which is the cysteine dependent aspartate directed protease, it actually gives you a very clear idea why these proteases are called as the caspases. So let us discuss this in a little bit detail. So the first thing is that these particular proteases, they are cysteine dependent. That means that their activity is dependent on the cysteine amino acid. If you look at the active site of these caspases, they have got this cysteine amino acid, and this cysteine amino acid in the active site of these proteases is essential for its activity. That means that if there is any mutation at this particular site, the caspases usually do not function normally. Therefore, we say that they are cysteine dependent. The aspartate directed means that if you look at the cleavage site of these caspases, they actually cleave a target protein only after an aspartic acid residues. That means that the aspartic acid is actually providing a direction or a cut site to these particular proteases. Therefore, they are called as the aspartate directed proteases. So cysteine is in their active site and they actually cleaves their target protein only after an aspartic acid residues. If you talk about the cut site of these caspases, as I've told you, they actually cut at a sequence which is called as the DEVD. Now this D is actually for the aspartate, this E is for the glutamate, this V is for the valine, and this D is again for the aspartate. So if you have got this sequence in the target protein, in a particular protein, this DEVD, aspartate at this site and aspartate at this site, these caspases are actually going to cut this particular protein at this particular site where the aspartate is present and at this site where the aspartate is present. So the aspartate is actually providing a direction for the these caspases to cut a particular protein. Therefore, they are called as the aspartate directed. So they actually recognize this sequence, the DEVD, aspartate at this side, aspartate at this side, and they cut at both sides of these aspartate. If you talk about the caspases, the initiator caspases, if you talk about the initiator caspases, the caspases are actually divided into two classes. One are called as the initiator caspases, and the examples are the caspase 2, caspase 8, 9, and 10. And the other class of the caspases, they are called as the executioner caspases, which are the caspase 3, the caspase 6, and the caspase 7. Once the apoptotic signal, if you summarize this, so the apoptotic signal, when that is received, this apoptotic signal are actually responsible for the activation of the initiator caspases, a very small number of them. And this small number of the initiator caspases are going for the activation of a very large number of the executioner caspases. And these are actually these executioner caspases which are responsible for the process of the apoptosis. If there is an accidental activation of the caspases, you only activate caspases when you are required with the process of the apoptosis. But if there is accidental activation of the caspases, you should have a backup plan for that. So the accidental activation of the caspases, that is prevented by a family of the proteins which are known as the inhibitor of the apoptosis, and their name is crystal clear. They are the inhibitor of the apoptosis. They are not going to allow the process of the apoptosis. Now the best characterized member of the inhibitor of the apoptosis family is the Iep Tree. This is one of the member of the Iep family which has been best characterized, and this Iep Tree is actually binding with the caspase 9, the caspase 3, and the caspase 7. And when the Iep Tree bind with these caspases, they inhibit these from performing their function. They are actually inhibiting their functions, or they are actually targeting these caspases for the degradation. So if there is an accidental activation of the caspases, these inhibitor of the apoptosis are actually going to stop the process of the apoptosis. Now, if you need apoptosis, that means that you have to get rid of these inhibitor of the apoptosis, because if these inhibitor of the apoptosis, they are there, you will not be able to activate the caspase 9, which is actually an initiator caspase. And if you are not able to activate the initiator caspase, of course you will not be able to activate the executioner caspase like the caspase 3 and 7, and you will not be able to go for the process of the apoptosis. Now that means that if you are going for the process of the apoptosis, you have to get rid of these inhibitor of the apoptosis. when the apoptosis process is needed and the BH123 aggregate forms, these aggregates, when they form, as I've told you in the last video, they are going for the outer membrane permeabilization of the mitochondria, and the cytochrome C, they are released. But when these BH123 aggregate, they are formed, they are not only responsible for the release of the cytochrome C, but they are also responsible for the release of the IAP inhibitors. That means you are actually releasing the inhibitors for the inhibitors. These are the inhibitors of the apoptosis, and they are actually stopping the process of the apoptosis. But when you stop the activity of these inhibitors, that means you can actually go for the process of the apoptosis. So the BH123 aggregates, they not only release the cytochrome C from the mitochondria, but they also release the IAP inhibitors. And when you inhibit this IAP, that means you can actually activate the Caspase 9, and you can actually activate the Caspase 3 and the Caspase 7. Now, when these executional caspases, as I've told you, they are actually the caspases which are responsible for the process of the apoptosis. So what are the targets for these executional caspases? Now, one of the important targets for these executional caspases, for example, the Caspase 3 or the Caspase 7, that is an inhibitor of the DNase. Now, you all know that DNase is an enzyme which is responsible for the fragmentation of the nuclear DNA. In normal condition, when there is no apoptosis, the action of this DNase is actually inhibited by an inhibitor. So when the executional caspases, when they are activated, what they do is that they are going to degrade the inhibitor of the DNase. And when the inhibitor of the DNase that has been degraded, that means that the DNase is now free to act, and this DNase will be responsible for the fragmentation of the nuclear DNA. So one of the important target of the executional caspases is actually to degrade an inhibitor of this DNase, allowing the DNase for the fragmentation of the nuclear DNA. Another important target of this executional caspase are the nuclear lamins. So when you talk about these nuclear lamins, they are actually proteins. They are actually made up of the intermediate filaments and associated protein, and these nuclear lamins are very important to provide a mechanical support to the nucleus of the cell. So when the executional caspases that degrade the nuclear lamins, that means you have broken down the mechanical support, and when you have broken down the mechanical support, you are actually going to lead for the fragmentation of the nucleus of the cell. So the nucleus of the cell has now been fragmented because the nuclear lamins that have been degraded. Another important target of the executional caspases, they are the cytoskeleton proteins. Now these cytoskeleton proteins, they are very important for the normal structure of the cells. So when the executional caspases, they cleave the cytoskeletal proteins. For example, the important cytoskeleton proteins in the cell are the actin, the myosin, and the tubulin, etc. So when the executional caspases, they cleave these cytoskeleton proteins, it actually leads to the disruption of the cytoskeleton. Another important thing that happens because of the degradation of the cytoskeletal protein is that there is membrane blebbing. There is cell membrane blebbing. Blebbing is actually the bulging of the membrane, and when you have degraded the cytoskeletal proteins, you have went for the membrane blebbing that actually leads to the fragmentation of the cell. Another important target of the executional caspases, that are the Golgi matrix proteins. Now the executional caspases that also cleave the Golgi matrix proteins. For example, one of the important protein in the Golgi matrix protein is the Golgi microtubule associated protein 120. For short, that is called as the GMAP 120. So when the executional caspase that lead for the degradation of the Golgi matrix protein, that lead to the fragmentation of the Golgi apparatus. That means that the executional caspases, they are actually going for the indiscriminate degradation of the proteins in the cell. They are degrading all of the kind of the proteins, thereby degrading the cell. Now, an important term that is used when you talk about the apoptosis is the apoptotic bodies. So what are these apoptotic bodies? Now, these apoptotic bodies, they are actually a vesicle containing parts of the dying cell. You are degrading the cell, that means you are disintegrating their parts. So apoptotic bodies, they are actually a vesicle containing parts of the dying cells. And these apoptotic bodies, they are usually made during the execution phase of the apoptosis. That means you have once you have activated the executional caspases, you have executed the apoptosis, and during this execution phase, when the cytoskeleton breaks up and there is membrane blebbing, as a result of the cytoskeleton breakup and the membrane blebbing, these apoptotic bodies, they are made. So these apoptotic bodies, they actually contain parts of the dying cell. Now, these apoptotic bodies, they are actually engulfed by the phagocytic cells, and when these apoptotic bodies, they are engulfed by the phagocytic cells, actually their components, they are recycled and they can be used for the synthesis or the formation of the new cells. We will continue the extrinsic apoptosis in the next video.